Hello, everybody. I'm James Gilbert. I am at the University of Hull. I'm interested in insect behavioural and nutritional ecology, particularly parental care behaviour. Our project, if you could move on to the next slide, is called Nutrigenomics and the Resilience of Bees in a Changing Climate. And it's co-supervised by Elizabeth Duncan at Leeds, who's interested in insect developmental biology and genomics. It's also co-supervised by Laurie Lawson-Handley, who's someone you'll meet later in this webinar because she's also presenting one of the other projects. So it is a cross-institution project. So two of the biggest threats to biodiversity are climate change and human-altered landscapes, but it's really rare to get a chance to study both of these effects acting together. And that is what this project aims to do. And we are going to be using solitary bees as our study system. So we all know that bees are super important for our food security because they pollinate our crops, but they're dependent on pollen from the landscape for their nutrition. And like many animals, many bee species are under threat from factors like climate change and changes to the nutrition available in the landscape. But we know surprisingly little about bee nutrition because most studies look at social species, which are in fact only a tiny minority of bee species. But in those social species, it's really difficult to study the larvae in isolation from the colony. So we know loads and loads about adult nutrition, but very little about the larval nutrition. And that's why we study solitary bees, because they have a simple manipulable system where we can study the nutrition of individual larvae. In my lab, we use a powerful technique called nutritional geometry, which can experimentally identify the optimal diet for a growing animal. And then you can effectively ask the animal which diet it prefers. And we've shown that the larvae of these mason bees that you can see here on the left grow largest when they eat the most carbs. You can see up the y-axis there. What we now want to know is how those effects play out in a warmer future. And Liz's lab at Leeds has identified genes that are up and down regulated in response to different nutrition, specifically those related to development of caste. And what we want to do is to identify those genes expressed in response to different combinations of diets and temperatures to get an idea of how a warming climate will affect the way bees use nutrition. So what you'll do, if we could go to the next slide, please, what you'll do is to put out bee cocoons at Hull, which will emerge and breed in our experimental nests. And those nests are set up so that you can extract the larvae and transfer them to individual nests with an artificial diet that we've formulated. You'll then put those into culture chambers set to different temperatures and monitor the amounts that they eat and excrete. And at the end, you'll assess their body chemistry to determine nutrient storage. And then working with Liz, you'll use high throughput sequencing to establish genome-wide expression profiles in response to these different treatments to establish the effect upon the bee's physiology. And this is going to be some of the most in-depth work on bee nutritional ecology to date. I find it really, really exciting. It's going to open up a huge number of questions across loads of different fields, all the way from agriculture and conservation to social biology and ecology. So if that sounds interesting and exciting to you, then please do get in touch because we'd love to hear from you.